Here we are at special senses. This is part 1. You can see this image here. We're looking at taste and smell and vision, and they're showing your somatic senses. So this is from the rest of your body, like touch. And then we have hearing, and we have equilibrium, and they're showing all of these messages going through the thalamus, and then where they're deposited within the brain. So touch is one of the general senses. It's mediated by general receptors. We covered this in chapter 13 already. So I'm going to kind of skip through the touch part. We're going to talk about vision in part 1. These two are going to be part 2, and these two we're going to talk about in part 3. Just to make sure we know some of these definitions, do we remember what a receptor is? So this is any structure that's specialized to detect a stimulus. So we've already seen receptors before, like we had an acetylcholine receptor. And what bound to the acetylcholine receptor was acetylcholine, right? Receptors are proteins. So they're made by the cell and expressed on the surface. What is a sense organ? A sense organ is nerve tissue that's surrounded by other tissues that enhances the response. So we're going to look at the anatomy of the eye, and then we're going to talk about the physiology of vision. But I wanted you to see this. I think it's pretty important that 70% of all of our sensory receptors are in the eye. That's saying all, right? Your entire body, the majority of them are in your eye. Half of the cerebral cortex is involved in processing visual information. So this should tell you how important your eyes are. Also, do you remember that we have 3 cranial nerves? And these 3 cranial nerves are controlling eye movement. So that should also tell you it's pretty important. We're also going to protect the eye with fat and the bony orbit, right? So remember, we have our frontal bone coming down on the inside of the eye, on the side that you can't see. We have part of the sphenoid, right? We have part of the ethmoid, part of the maxilla. So several bones come together to form your eye socket or your bony orbit. Accessory structures of the eye, I have them listed here. Eyebrows. One of the things is your eyebrows are supposed to be nice and bushy, right? This stops sweat from dripping down into your eye. Obviously, we have eyelids that can close in case somebody's trying to poke us in the eye, right? Conjunctiva, I'll show you in just a second. We have a lacrimal apparatus, and then, of course, we have these 6 extrinsic eye muscles. So eyebrows prevent sweat from reaching the eye. Eyelids, they're going to blink, offers protection from foreign objects. We have something called the conjunctiva. You might have heard of conjunctivitis, right? But this is a mucous membrane that produces a lubricating mucus secretion. Keep your eye moist, right? We also have the lacrimal apparatus. So this is the lacrimal gland and the ducts that drain into the nasal cavity. I have an image coming up. And then we have our 6 eye muscles that allow us to track objects, maintain the shape of the eyeball. Here's your conjunctiva. So you can see that this transparent membrane does like a little infolding. And this mucous membrane is going to lubricate the eye. You can see there'd be some nice little pockets that bacteria could kind of stay in and multiply in. Here's our lacrimal gland, and you can see these lacrimal ducts, and they produce tears. And the whole point of tears are supposed to be to come across the eye, clean the eye, get any foreign object out of the eye, and then they're going to go through these lacrimal canaliculi. Canaliculae for 1, canaliculi for 2, come through and drain into the nasolacrimal duct and then eventually into the nose, or can go behind to your pharynx. And here we have our 6 eye muscles, and you can see that they're attached to the eye and they allow for us to move the eye in many directions. And then some of them are actually used to anchor the eye in the bony cavity. So you can have conjunctivitis, which is an inflammation of the conjunctiva resulting in some sort of redness or irritated eyes. You could have an inflamed sebaceous gland. That's a stye. That's when your eye has like your eyelid gets red and puffy and there's fluid in there. It's usually some sort of blocked sebaceous gland in your eyelid. Remember, you have hairs, you have eyelashes coming out of your eyelid, so the sebaceous glands could get clogged. And then more commonly we think of pink eye, right? It's really just conjunctivitis. That's the inflammation, but it's because you have some sort of infection, bacterial or viral. We're going to move on to the structure of the eyeball. So the wall of the eye contains 3 layers or tunicas. We have a fibrous layer, a vascular layer, and an inner layer. And we also have some cavities that are filled with fluid, and we call them humors. And we'll talk about the vitreous and aqueous humor in a moment. So the lens separates the internal cavity into anterior and posterior segments. You can see here they've peeled away the layers so you can see them more clearly. So here's the fibrous layer, the pink is the vascular layer, and then this yellow is going to be the inner layer. So we have the 3 layers, right? Fibrous layer, vascular, and inner or internal layer. And then we're looking at the structures that are housed within each layer. So in the fibrous outer layer, we have the sclera, that's the white of your eye, and this is also where your muscles are attached. We also have the cornea. This is the thing that sits directly on top of the colored part of your eye. Then we move to the vascular layer. So we have something called the choroid, which is basically blood vessels. We have ciliary bodies, which are going to help control the lens shape. We have the colored part of the eye, which is the iris, and we have the opening, which varies the amount of light that can come into the eye. That's the pupil. On the internal layer, that was the yellow in that image, we have the retina, and the retina has rods and cones, and then all those messages are going to converge on the optic nerve and send the message to your brain. Hopefully you remember this is cranial nerve number 2. First we have the fibrous layer. So this is the cornea, and all of this is the white of the eye or the sclera, and this is where your muscles are attached. So I'm going to clear that, and then we'll do the vascular layer, everything here in red. So the iris is here, that's the colored part of the eye, and these little finger-like projections with strings to the lens of the eye, those are the ciliary bodies. And the ciliary bodies can make the lens shorter and fatter, or they can pull it apart and elongate it. And then all of this, once we go past the ciliary bodies, all of this structure is called the choroid, and basically this is the blood supply to the cells of the eye. And that leads us to the inner layer. So the inner layer is the retina and the optic nerve. So this yellow sheet here, we're going to have cells, and those cells are rods and cones. We'll talk about those in a minute. And all of those rods and cones are going to converge. There's several synapse events happening here, and then we converge into the optic nerve. Since we're talking about the iris, so there's the colored part of the eye, and there's the pupil, there's the opening, I wanted to show you the parasympathetic and the sympathetic. So remember, parasympathetic is rest and digest, and sympathetic is fight or flight. So why does this make sense? What happens during rest and digest? We said it's for close-up vision, right? So the best example of parasympathetic is you've just eaten a meal and you're sitting in a chair reading, right? You're going to constrict the pupil. You don't need as much light. Well, guess what we're going to do if you're trying to run away from something? You need to dilate the pupil and get as much light into the eye as possible so you can see to run away. So you can have a condition called retinal detachment. This is where the pigmented and the neural layers are going to separate. It can lead to permanent blindness. It can happen due to some sort of traumatic blow to the head or a sudden kind of jerking, stopping of the head during movement. So here, if the retina peels away from the choroid, that's retinal detachment. And the only thing that's keeping the retina pressed firmly against the vascular layer, the choroid, is all of this fluid. That's what they were trying to show you with this white translucent kind of material. This is called your vitreous humor. So the pressure of this fluid is what actually keeps the retina attached to the vascular layer, which is the choroid. That leads us to the fluids. So we have vitreous humor, posterior cavity filled with a clear gel. It transmits light, holds the retina firmly against the pigmented layer, and contributes to the interocular pressure. The aqueous humor is the anterior, so usually A and A is how I remember this, is filled with a clear fluid similar to blood plasma. It's going to supply nutrients and oxygen to the lens and to the cornea while carrying away waste. And then finally we have the lens, which is avascular, so no blood supply. We should know that term by now. No direct blood supply, right? It gets it from the surrounding tissue. This is a concave, transparent, flexible structure that can change shape and allow for you to focus the light. So here's the cornea, this structure here. Then we have aqueous humor. So this is aqueous humor. It's in the anterior cavity. Here's your pupil. This is your iris, the colored part of your eye. And now we have the lens. And then we have vitreous humor in the posterior segment. So what's going to happen is light's going to enter the eye. It's going to pass through the cornea. Then it's going to pass through the aqueous humor. Then it's going to pass through the pupil. It's going to hit the lens, and the light's going to be reflected through the vitreous humor to the back of the eye onto the retina. You may have heard of glaucoma. This is where the drainage of the aqueous humor is blocked, and it causes fluid to back up and increase the pressure within the eye. If you have increased pressure, it can actually compress the retina and the optic nerve and lead to blindness. So now let's move into the physiology, right? So the eyes are only going to respond to visible light, so you have to be able to see light being emitted or reflected from an object in the environment. Light must cause a photochemical reaction. So basically you're taking light and you're turning it into a chemical reaction in order to produce a nerve signal, that should be telling you action potential, that our brains can notice. The lenses of the eye can also refract light because they are curved on both sides. The optical components. These are the structures that emit light, refract light, and focus the image on the retina. So again, light is coming in, right? It goes through the cornea, it goes through the aqueous humor, it goes through the pupil, and it bounces off of the lens, or refracts off the lens, goes through the vitreous humor, so this is vitreous humor, and the light goes all the way back here to the retina. The retina and the optic nerve are the neural components, so they're not transmitting the light, they're converting the light. So once the light goes through all these structures and hits the retina is going to take the light and it's going to turn it into an action potential. So we're going from light to a chemical reaction. And then all of the signals from the retina are going to merge onto the optic nerve, and then we're going to go to the brain. Could you explain to me how light is focused on the retina? Could you go from light through all the structures of the eye and where the light image is being reflected or refracted onto? So here is my cornea, then I go through aqueous humor, then I go through the pupil, then I hit the lens, then I go through the vitreous humor, and I come back here to the retina. The retina will then convert the light energy into action potentials, and we call that phototransduction. The photoreceptors are called rods and cones, and they're modified neurons. So the rods are what we're going to use in dim light, no color vision, or sharp images. So this is sometimes referred to as night vision, and cones for bright light, this is sometimes referred to as day vision. So there's an area that contains mostly cones, the macula lutea, and then there's the fovea centralis. This is a tiny pit at the center of the macula lutea, and that contains all the cones. So this is where we have the best visual acuity. What they're trying to say is this is where we can focus light to do small, detailed tasks that require a lot of vision. Here's the area that has lots of cones in it. Here's the fovea centralis. So this is the area that we're going to focus the light when we're trying to do small, detailed work. There's also something I want to show you is the optic disc, sometimes called the blind spot. So you see how all of this is retina. So all of the yellow is retina, but you see how there's no retina right here in that spot? That's called the blind spot. So if light happens to hit that area where there is no retina, there's no image that's going to be produced. So if you're looking at an object, you're going to have an area that is your blind spot. But what your brain does is your brain looks at this part and looks at this part and fills in this part so that you don't have gaps in your vision. You know, imagine if you're looking at a TV screen and a bunch of pixels have gone out, that's annoying, right? So your brain copies and fills in. If you want to, you can Google this. So go Google blind spot tests and you can do some of them. It's kind of interesting. So we have enlarged this area. So here's still the retina, here's the choroid, and these are all the cells that are going to synapse. So what's going to happen is the light's coming in, right? So we went through the cornea, aqueous humor, the pupil, the lens, the vitreous humor, and we said we went to the back of the eye. So the light is coming here to the retina. The light has to come all the way back here. It has to excite these cells, and then a chain reaction of events occurs, and they all merge to form the optic nerve. So the axons, these are all axons of this last neuron, and all of those axons merge to form the optic nerve. We enlarge again. So here is, all of this is retina, right? Here, back here is the pigmented layer. So back here you're going to have the choroid. These are your rods. You can see why they're called rods. And these are cones. So what happens is the light goes all the way to the back. It excites the rods and cones. The rods and cones are going to release their neurotransmitter. They're going to synapse with this cell in pink. This is called a bipolar cell. And then the bipolar cells are going to synapse with the ganglion cells. And then you can see that all of the ganglion cell axons are going to merge, and all of this message is going to be sent on the optic nerve to the brain. So the light has to come all the way in, and then it bounces back and then goes to the brain. Why do we have both rods and cones? You can see here that rods are for night vision and cones are for day vision. So single type of cell receptor is incapable of providing both high sensitivity and high resolution. So we just don't have that. So we have two depending on our needs. So think about night vision. A high degree of spatial, so they're saying you can see all of this wide space, but no ability to resolve detail. So the farther you are away, this gets fuzzy. So low detail, but high, so increased high degree of spatial. So you can see a lot of space in front of you, but it's not very detailed. And really this is just alerting us to motion. If we see something coming at us from the side, you know, something trying to eat us, we're going to try to get away, right? So cones are for daytime vision. So this says high resolution vision, but little spatial summation. They're saying you can see this much area, but you can see all of it very clearly. So this is why we have two different types of receptor cells depending on daytime or nighttime amount of light. If you look at the anatomy, how they're linked together, or the synapses here. Remember, here we have our rods, and we said they synapse with bipolar cells. And then the bipolar cells synapse with ganglion cells. And the ganglion cells are all the ones that merge to form the optic nerve. So I want you to see the difference between the rods and the cones. Do you see how each rod has its own line to a neuron? Right? There's no convergence. Its cone goes directly to its own bipolar, directly to its own ganglion, and then all of those converge to form the optic nerve, or the axons come together to form the optic nerve. What about the rods? Here you have 5 rods that are all converging on one bipolar, and then all the bipolars converge on one ganglion cell. So that's why this is low detail and this is high detail. Oops, sorry, detail. I did want to mention color blindness real quick. Basically, if you're lacking one or more cones, so there's different pigments. Remember, pigments are just proteins, colored proteins. I'm not going to get into the anatomy of the rods and cones and the pigments. It's a little bit more complicated, but basically you're missing one of the cone pigments, and it allows you to, or doesn't allow you to see certain colors. So the most common type is red-green, which either the red cones or the green cones are absent. So depending on which cone is missing, you can't see that color, right? So if you're missing the red cone, then your red is going to appear green to you. Usually people will train themselves and they'll learn the different shades of the color, and so they can tell whether it's green or red, but usually they have to learn this, right? And this is an X-linked condition. It means it's carried on one of the X chromosomes. So remember, if you're a female, you have 2 X chromosomes. If you give a copy that's missing a gene, so let's say you give them this X chromosome and it's missing the allele for the one of the cone pigments like red, then if it goes to a son, you're missing that allele, and then the son has color blindness. So it affects males more than women, than females. Next, I want to discuss focusing light on the retina. So we're going to do this differently whether we're doing distant vision or close-up vision. So for distant vision, your ciliary muscles are completely relaxed. It causes a pull on the ciliary zonule, and as a result, the lens is stretched flat. So really, if you want to say the ciliary muscles are relaxed and it causes the lens to stretch flat, that's what we need for distant vision. Here's the image, the item we're looking at. We can see that that image, we're going to have light hitting it, right? And the light is going to bounce off the image. It's going to go through the cornea, through the aqueous humor, through the pupil. It's going to hit the lens, and it's going to bounce to the back of the eye, going through the vitreous humor, right? And it goes to the retina. And the image is inverted on the back of the eye. And in the retina, this is where we have the cones and the rods, where depending if it's day or night, and we're going to take this photo light message and we're going to generate an action potential from it. So you can see that the ciliary muscles are relaxed. They're pulling on the zonule, and that is causing the lens to get flat. So stretching the lens out. Focusing for close vision, we have to make some adjustments. We have to change the lens shape. We have to constrict the pupil, and we have to rotate our eyes a little bit towards the object being viewed. We can see that we've moved the item closer. So we still have to have light bouncing off of this object, going through the cornea, the aqueous humor, the pupil, the lens. But what do you notice about the lens? You see how the ciliary muscles are coming closer together, right? And you're going to see the lens become kind of shorter and fatter, not the elongated version. And now the inverted image makes it to the retina. So this has to do with refracting light. I really just want you to follow kind of the basics that are on here. The textbook goes into much more detail about how exactly this bending pattern or refraction of the light works and how it bounces off the lens. But remember that the image has to make it to the retina, because the retina is where you have the rods and the cones. I'm sure we've heard of people being nearsighted or farsighted. So if the eyeball is too long, the focal point is in front of the retina, so you can't see as well. Things look fuzzy, right? You can have farsightedness when the eyeball is too short. The focal point is behind the retina. So we're going to have glasses. In either way, we're going to have to put something in front of our eye in order to make the refraction of light turn the image or place the image directly on the retina. You can also have astigmatism. So this is when you have unequal curvatures of the cornea or the lens, and this is going to cause the light to not bounce properly or refract properly, and it's going to cause image problems with the image hitting directly on the retina. I wanted to show you some images. So here's a normal. So the light comes in, it refracts off the lens, and it hits the focal plane. So our image is right here on the back of our retina. Our rods and cones can turn that photo or light into an action potential, do that photochemical reaction, and then we can carry the message, the action potential on the optic nerve, and we can take it to the brain, and we can process what we're viewing. So what happens if you're nearsighted? So myopic eye, your eyeball is too long. You can see they've stretched kind of the eye. So what happens when light is shining through the cornea, the aqueous humor, the pupil, the lens? The image ends up here. Your focal plane is too far away from the retina. So this makes things kind of fuzzy, right? You might still get an image up here, and that'll be okay. You can see this and you can see this, but you just can't see the majority of it. So what do? We put a corrective lens in front of your eye. So now we're changing the refraction point and we're causing the image to go where it should, which would be the retina. So what happens if you're farsighted? Hyperoptic. You can see here that the eyeball is too short. It's kind of squished a little. So as the light comes through, you're actually putting the image all the way back here. It's behind the retina. It's not going to do you any good, right? So then we put a corrective lens in front. We correct the refraction of the light, and it ends up right here, the focal plane where it should be, right on the retina. The axons from the ganglion cells form the optic nerve. The medial fibers from each eye cross over at something called the optic chiasm, and then they continue as optic tracts. Eventually, they're going to your visual cortex in your occipital lobe. So here's your right eye, here's your left eye. We have our optic nerve. Here's that chiasm crossover, but let's just follow this side. So you have the optic nerve forms the optic tract and goes back here to your occipital lobe where you have your visual cortex. You can see the left eye does the same thing back here. So what happens with some of the nerve fibers? So here is the right eye, and we can see the optic nerve and the optic tract crosses over and comes over here to this side. So here we go right side and we send the message to left side. We also go right side and send the message to the right side. What do we do on the left? We have some of the message that crosses over the optic chiasm and goes to the right, and we have some of the message that comes over here and goes to the left. What's interesting about this is you can look at the field of vision. So we're saying from right eye here, it says right eye only, but follow right eye all the way over. So this is left eye only, so there's no right in this tiny little section. So your right eye commands this entire field. This is like 75% of your total vision is your right eye. Follow your left eye. Your left eye is all of this. 75% of your field of vision is processed by your left eye. This is right eye only, so that means there's no left. So there's a crossover, right? So your right eye can see from here to here, and then your left eye can see from here to here. So all of this is overlapped. This probably makes sense because what happens if you lose right eye? Something happens to your right eye. What can your left eye see? Your left eye still sees 75% of your field. What's the only thing you won't be able to see is kind of this right side peripheral. What happens if you lost your left eye? You'd still be able to see 75% of your field of view. You just would not be able to see this tiny section or left peripheral. So we have a lot of redundancy with the eyes. That should tell you how important it is.